Great. So my name is Jeff Iliff. I'm currently at Oregon Health Science University. I actually just arrived here in this last December, but for the prior 3 years, I'd been at the University of Rochester in Mike and Niedergart's lab, same place where Ben, Kress, and Harris are, working in a lab that is best known for glial cell biology and disease states such as stroke and traumatic brain injury, Alzheimer's disease, and those sorts of things. But through the course of my project there, we ended up sort of stumbling into the realm of CSF dynamics, which is something actually that none of us knew anything about. So to me, it's kind of nice to get to talk to you guys, hopefully now and maybe in the future, to figure out if there's ways where our work could move forward more in the context of modeling and those sorts of things. So it looks like Harris is online now. I'm going to have him introduce himself real quick because some of what he's doing right now is fairly relevant to, I think, what a lot of you are doing. Harris, are you there?
Yeah, can you hear me?
Yeah. So introduce yourself, Harris.
Hi, I'm Harris Weber. I'm an undergraduate here. Doing some work with Jeff, working with the spinal cord, trying to characterize some of the system dynamics going down there.
All right. So I've never taken part in a GoToMeeting like this, but if at all possible, if you guys are willing to interrupt and interject, I would love that. I would love this to be more of a conversation than me just spouting 50 minutes worth of data at you. So feel free to interrupt me along the way. The title of the talk that I'm going to give it's called Defining Parenchymal Pathway of CSF Circulation. And it encompasses some published data, some data that's under review right now, but also I'm trying to get at what I'd like to leave you with is a broader view of what CSF may be doing and where it may be interacting and how that might influence and maybe should influence our view of CSF dynamics, but also neuropathology, which is sort of the angle that we come to it from. We're much more of a cellular neurobiology lab than a modeling lab or not even a CSF lab. So the talk will have a decidedly cellular neuroscience event to it. So, anything to add, Brendan, before we get going here?
No, it sounds great. I have two thumbs up, thinking of all the things we're going to hear about.
Okay. So, one quick question is, if I try to play a movie, is it going to work?
It should work.
Okay. Well, we'll find out. All right. So I wanted to start. Thankfully, this is actually a subject with this crowd I probably don't have to talk much about, which is sort of nice, and that's the CSF circulation. So it's regarded as the third circulation. So the first is the blood circulation, the second is the peripheral lymphatic system, and the third is this sort of oddball fluid system in the brain, whereby fluid is secreted, this kind of very salty, non-proteinaceous fluid is secreted four plexus deep within the brains, deep within the ventricles of the brain, in the third, lateral, and fourth ventricles. That column, this is sort of the textbook view of CSF dynamics that you might have gotten in grad school or med school or beauty school or whatever school you went to. So the CSF is secreted in the ventricles, and then is regarded to flow in sort of a continuous column out through the ventricular system, spilling out into the cisterna magna, and then wider cisterna subarachnoids, where it basically surrounds the brain and also the spinal cord. From there, the CSF is thought to be reabsorbed. When you talk to clinicians, where they think it's reabsorbed is typically through the arachnoid granulation in the dural sinuses, through these sort of valve-like structures. So if you talk to people in the field, you hear a lot more about reabsorption through the cervical lymphatics after the CSF has made its way out along cranial nerves, like the the optic nerves or the cribriform plate, or perhaps even spinal nerves down further down along the spinal cord, ultimately giving the CSF access to the peripheral interstitium, the peripheral lymphatic system, and then ultimate drainage back into the blood circulation. So it's this loop where CSF originates from the blood at the choroid plexus. It moves through this system out into the subarachnoid space, and ultimately, one way or the other, back into the blood. Now, the function of CSF is, when you look it up on Wikipedia, you get sort of three main functions, right? So one is buoyancy. The brain floats in this volume of fluid. So instead of having, even though it has a mass of over a kilogram, it has sort of an effective weight because it's floating of like 25 grams. So structurally, it's supported in that way, and you can afford to have a big, gooey mass that doesn't, smush down, and that's able to sort of maintain its physical structure. This layer of CSF that surrounds the brain and surrounds the spinal cord is thought to provide some level of physical protection from external blows. And then there's this third function of CSF that's regarded as an excretory function, or more, depending on where you read, more of a sort of an equilibration function. So the sense of this is that because CSF is inside the brain in the ventricles, and it's outside the brain in the subarachnoid space, somehow or other this acts as a sink to different metabolites that are inside the brain, allowing things in the brain to sort of equilibrate through the space. And in that process, different solutes, particularly waste solutes that are present in the brain interstitium, sort of make their way out of the brain into either the internal ventricular space, the external subarachnoid space, and then eventually out of the brain. So it's a way, perhaps, for the brain to get rid of metabolites in space that it can't get rid of across the blood-brain barrier or any other way. Now that's reflected in the field of neurology by the use of different biomarkers for disease that are found in the CSF. So an emerging thing in the field of Alzheimer's disease research is, looking in lumbar CSF at levels of amyloid beta or tau or phosphorylated tau, measuring the amounts of those that are in the CSF and deriving some ratio of these that is predictive of the onset of Alzheimer's disease and the progression of Alzheimer's disease. Now, the fact that these are biomarkers indicates the fact that these are all interstitial solutes. In fact, tau is an intercellular solute. So this is in cells, particularly in neurons of the brain, tau is. And so here you have cells that are present in the brain interstitium or in cells within the brain that somehow or other they make their way out to the CSF spaces where they can be sampled and used as a biomarker. So they're sort of implicit in this, in the status of these as biomarkers, is this excretory role of CSF for the wider brain parenchyma.
So actually, Jeff, can I ask a question?
Yeah.
So you said that the tau protein would be predictive of the amount of Alzheimer's onset.
Mm-hmm.
So I saw that there's a recent paper looking at that, but I just wanted to know, is that really confirmed or is that a feeling?
It's beginning to be more than a feeling. So it's in the land between someone's idea and clinical practice at this point. So there have actually been a number of studies now that have validated specifically ratios of phosphorylated tau and amyloid beta 42 in Alzheimer's disease, and it seems like the most predictive one so far is the ratio of AB42 to phospho-tau. AB42 goes down precipitously as Alzheimer's disease progresses because it's sticking inside the brain, and phospho-tau actually it seems to increase. So the ratio drops because the phospho-tau is in the denominator and amyloid beta is in the numerator. So it's increasingly being validated.
Okay, thanks.
All right. So the problem is if there's an excretory function of the CSF, you have, especially larger brains like the human brain, you have sort of a space, or you have a distance issue. So if it's diffusion that's accounting for that movement, the distances that solutes must diffuse in the human brain become somewhat prohibitive. So in the human brain, the distance between some parts of the interstitial space and either a ventricular or subarachnoid CSF compartment are up to a centimeter. And depending on the size of the molecule, it could take an awful long time for that diffusion to occur. In the case of urea, which is very small, the time for diffusion, that distance is about 5 hours. But in the case of albumin, which is a fairly good-sized protein, which is incidentally about the size of tau, the diffusion time is in the hundreds of hours. So this is a very slow way for particularly large solutes to get to the CSF. And it seems as though, just intuitively, that's not really efficient enough for an organ as sensitive as the brain and the wider CNS is, for it to be clearing its way. From this fact came in the 70s, 80s, and 90s this idea of bulk interstitial flow. So what this is the notion that the brain interstitial fluid, which is all the fluid in between the cells here of the brain, is not static, but rather flowing through the tissue like water flows through an underground aquifer, right? So it's the fluid medium itself is flowing all the time. And with it, it's taking all of the solutes that are in it. So albumin and urea, in this case, would flow with the fluid, interstitial fluid, independent of their size. So large molecules would flow in this river at the same speed as smaller molecules. And when calculations have been done looking at sort of gross rates of bulk flow in the rat, you end up with a half time of turnover for the interstitial fluid being about in the order of 10 hours or so. When you compare that to the rate of CSF turnover within the ventricular system, you see that it's quite a bit slower. And on the order of 1 to 2 hours for rat CSF. So this process seems to be a pretty slow process, and even in this current conception, it's thought to be a fairly diffuse process. So interstitial fluid is just sort of moving vaguely through the brain from wherever its source is, and the source could be either metabolic water produced by the brain or water coming across the brain microvasculature out to the CSF spaces. So it's this diffuse process of movement, which seems a little bit strange because we think of the brain as a fairly high-performance organ. It's metabolically very active. It's producing a lot of waste, and yet it's exquisitely sensitive to changes in its interstitial environment. And so the brain that is so highly involved in so many other senses doesn't appear to have an anatomical pathway to facilitate a more rapid and efficient clearance of interstitial waste, when in comparison to skin, which is, we think from a CNS perspective, comparatively primitive and simple, right? It has a very complex second vasculature to do this. And so that question is what motivated all the work that I'll be talking about today, of whether there's some anatomical substrate, some way that the brain has organized this set of flow to make it more efficient. So to begin to answer that question, one of the first things that we did was to find out whether CSF...
So Jeff, can I ask a question? Sorry to interrupt again. On that ISF flow number, that would mean if you took it for the whole brain, I just did a little back of the envelope, it's about 400 milliliters per day for a human.
For the human brain?
Yeah. So if you take that ISF number there, and you just calculate it for 1,400 grams of brain tissue, you get about 400 milliliters per day.
So that's, okay.
So that's a lot more. I mean, you're saying it's 4 times as much as, well, 3 times as much as the baseline. 125 is the normal quoted number of turnover.
So this is for, so this number here is calculated for rats, right?
Yeah, I'm just saying if it was similar for human, right?
Oh, okay. Yeah, I don't, to my knowledge, that study hasn't been done in humans, and whether or not these rates scale for humans, I don't know that anyone really has a concept of that.
And the second question is about this distance to the CSF surface. Is there a reference you have for that? For 1 centimeter?
I can look one up for you.
So I've never seen exactly that, and I'm wondering, what was the approximate surface area?
I think what this is, so this is referenced out of Helen Sayre's, she had an article in Physiological Reviews where she outlined the concept of interstitial bulk flow originally in 1971. This value of 1 centimeter and these calculated diffusion times are taken from her work here. So I can look into there and see if there's a reference to this particular distance. My assumption is this is just sort of taken from neuroanatomical work of how far is what's sort of the greatest distance from any particular place in the brain to a CSF structure.
That's interesting for modeling purposes to know, how that would correlate to the total surface area, because that means there's tons of surface area.
Yeah, I think the implication of that is that, yeah, there would be. What I would wonder is, and this wasn't referenced, sort of how much of the brain's volume is further from here? How much of it is closer to, what's the sort of distribution of proximity to CSF, and how does that influence things? Because that has a pretty big bearing on whether our studies in rodents are really transferable to humans, right? So if a lot, the more of the brain that is further from CSF spaces, in principle, more important bulk flow and organized bulk flow ends up being, right? But I don't know, I'm not necessarily certain, and maybe in your group there maybe know this better, how much that's been investigated. So where we started with the process was to look to see whether CSF in these different compartments was actually exchanging with the interstitial fluid. So this is very similar to some work that was done in the earlier 80s with some different tracers, where we sort of moved it into fluorescence-based. That's what our lab does, and that's sort of where the field is at right now. So we started with injecting different molecular weight tracers into the cerebral ventricles, and the lateral ventricle walls, and just looking to see how extensively they moved through the brain interstitium. And what we saw was more or less what we expected, and that is, in general, you inject a tracer into the ventricular system, you see that it gains access to the sort of paraventricular tissue. But it looks to be sort of a 1/r² distribution. It seems to be much higher near the wall of the ventricle, and then it seems to drop off as you move away from the ventricles. The smaller tracer, this is less than a kilodalton Alexa 594. It moves more broadly through the brain, but not all the way through the brain. You can see here it's largely dark. This larger tracer, 2 kilodalton dextran, that's in blue here, is more restricted. A very large tracer, this is a 2 million Dalton dextran. It really doesn't penetrate through the ependymal layer, but rather kind of lodges in these perivascular spaces along the CSF surfaces here. So, it was interesting, though, that when we shifted the tracer from the ventricle, the subarachnoid space of the basis system, or of the magnetic, we saw a very different. Traces that is small. Whereas the larger tracers, you can see they do exchange with the brain interstitium. They move back into the brain. The small tracer, you can see it's exchanging with the surrounding interstitium. The large tracer, it's confined to these what appear to be perivascular spaces. So this was sort of the surprising bit to us, and that was that a large proportion, what appeared to be a large proportion of the CSF in the subarachnoid space, actually moving back into the brain, and it seems to be doing so along these perivascular spaces. When we looked at higher magnification, what we saw, so this is in a reporter mouse line that we have that expresses one fluorescent protein in endothelial cells and another fluorescent protein in smooth muscle cells. And what we saw was that tracer that was injected into the basal cisterns actually moved into the brain fairly specifically along arterial pathways. So this is the cerebral cortex here. You see the tracer, in this case it's ovalbumin, which is a 45 kilodalton tracer, moves into the brain along these penetrating arteries and gains access to the basal lamina of the terminal capillary beds. This is an ascending vein here. You see that the tracer really doesn't enter along this venous pathway. So there's a pretty big difference, artery versus vein, as to whether or not the CSF is entering along that pathway. And when you look in the basal ganglia, you see something analogous but greater in magnitude. So this is the lenticular striate artery here. It's a large artery moving up to the ventral surface of the brain. You see that the tracer, a lot of the CSF is moving along this pathway. At higher magnification, you can see that it's moving along the basement membrane that surrounds the smooth muscles, the vascular smooth muscle. When you capture a large draining vein in an adjacent frame, you see that the tracer really isn't moving along this pathway. So there's, again, a polarization of CSF inflow into the brain along these arterial paths. So the next thing we wanted to do was to get a sense of the dynamics of that movement. So these are all sort of pulse chase experiments where you inject, you wait a period of time, you fix the animal, you slice it, and you look to see where the tracer gets, which is really no different than what had been done in the field up to the 1990s. So this is kind of an old school approach to looking at these sorts of things, and it doesn't necessarily give you a view of the dynamics of the system. So the next thing we did was we used in vivo two-photon imaging to try to catch the dynamics of this in real time. So I don't know how familiar people there are with two-photon imaging. The Cliff's Notes version is that it allows, it's an imaging approach that allows you to image fluorescence deeper into tissue than you could with something like confocal microscopy. So with two-photon imaging, you can image fairly routinely 400 or 500 microns down into living brain tissue, whereas with confocal microscopy, you're usually limited to about 50 microns. Now in a human, 500 microns isn't very much, but in a mouse, that's much of the depth of the cortex. So we're able to image a large hunk of the cortex sort of in real time. So this is a two-photon image taken from the surface of the mouse cortex. The vasculature is outlined with a fluorescent dextran that's been injected IV. So this is the surface arterial here, or artery here. This is surface vein there. 5 minutes after injection of small and large molecular tracer, you see that the tracer, which was in the cisternal compartment at the cisterna magna, it's now up at the dorsal cortical surface in what appears to be a subcompartment immediately surrounding these surface arteries here, right? So it's tracer in CSF moving along the surface very rapidly. These are images taken from 100 microns below the cortical surface. So these are penetrating arterioles. You see them in cross-section because they're diving sort of normal to the brain surface. And you see that the tracer enters initially along these penetrating arterioles, and then it moves out into the surrounding interstitium. And over time, you can see that it accumulates. Now, the large tracer, which is in green, and it kind of comes out as kind of greenish, bluish white here, it actually remains confined to these perivascular spaces and doesn't move freely into the surrounding interstitium. Those spaces are where we're imaging Robin spaces. So these are the big CSF-filled spaces that surround the penetrating vessels. But then from those spaces, small tracer is able to move out into the surrounding interstitium. Under higher power, you can see that more clearly. You can see that the large tracer, it's in these Virchow-Robin spaces, and then the small tracer moves out into the surrounding interstitium and exchanges fairly freely. You can conduct dynamic imaging and get sort of a time course. So this is the dynamic series taken from the cortical surface, and you see, in this case, it's a 40 kilodalton single tracer. Moves along these peri-arterial spaces, and then these are penetrating vessels. And you can do this imaging in a 3D tissue block. So these are images, a series of images acquired cortical surface and then at different depths below the cortical surfaces. And you see that the tracer, it begins along the cortical surface, but then it moves fairly quickly inward along these penetrating vessels, eventually exchanging with the brain interstitium. So this gives us a chance to sort of view this CSF interstitial fluid exchange in real time and to really see it in action, which helps us to have some confidence that it's actually happening, because we can, you basically see it before your eyes. We wanted to quantify how much of the CSF that's in the basal cisterns, that's in the cisterna magna, how much of that CSF is actually re-entering the brain. So is this 5%, is this 10%? How much of the CSF that's out there is re-entering the brain along those perivascular spaces and exchanging with the interstitium? So to find that out, we injected different radio tracers. In this case, mannitol, which is small, and dextran, which is larger, to 10 kilodalton dextran, into the cisterna magna. You wait a period of time, and then you count the amount of radiation that's entered the brain in that period of time. And you can see that, with a larger tracer, it enters the brain more slowly than the small tracer. So there seems to be some sort of size-based exclusion of molecules out of these pathways. But the small tracer, if you look at it, it's up to 40% of the mannitol that's injected into the cisterna magna enters the brain. Now, when we did these are counts only basically of the brainstem forward, and they don't include the spinal cord. So this is an estimate that's basically only includes the brain portion of the CNS. So it's probably underestimating the amount of CSF that's re-entering CNS parenchyma. The other thing that we think about is we see a size effect here. So the smaller we get, the more of this, the more the tracer gets into the brain. Now, mannitol isn't water, and water can enter the brain more readily than mannitol can. So we assume that this 40% is also likely an underestimate, and that CSF itself, at least the water of CSF, may be moving, even a larger proportion of it may be moving into the brain than 40%. But we don't necessarily have a good way of looking at that, so we haven't been able to do that. So that was how we began to define the pathway by which CSF entered the brain and exchanged with the interstitial fluid compartment. Our next question was how interstitial fluid left the brain. So did it follow a similar anatomical pathway as CSF when it was coming in? And so to do that, and here I'm summarizing probably a year's worth of work and scores of animals that were injected basically in 2 slices here. What we did is we injected different tracers. In this case, I'm showing ovalbumin conjugated Alexa 647. So that's a 45 kilodalton fluorescent tracer injected in the caudate nucleus. We waited 30 minutes, fixed the animal, slice it, counter-labeled GFAP so you can sort of see structure, and then look to see where it goes. And you can this is sort of a representative image of what we saw more generally. What we saw more generally was that tracer injected into the interstitium moved just sort of diffusely through the interstitial space, as you might in sort of a spherical radial pattern. When it encountered interstitial capillaries, you would see that the tracer would move forward along those capillaries much faster than it did through the bulk interstitial fluid, right? So if you looked under high power out ahead of these wave fronts here, you'd see that tracer would be way out ahead of the capillaries. So that would seem to be a permissive low resistance pathway for interstitial movement. And then when you saw that the interstitial fluid gained access to particularly large caliber draining veins, and here this is the cauda-rhinal vein, but then we also saw it along the internal cerebral vein, it would sort of jump out of the brain interstitium. So here you can see it sort of moving very quickly outward to the brain surface along these large caliber veins. And we saw this again and again. We saw it both lateral ventrally here, and we also see it medially along the internal cerebral veins. So there are very particular pathways by which interstitial fluid left the brain, and it seemed to be focused on these large caliber draining veins. So then
So Jeff, can I interject again? A question about that. So what level of impact is the inflammation having on the the movement there? I mean, would you guess that it's small or large?
Yeah, so these injections here were done acutely, so we don't expect there to be a lot of inflammation. So to do these, we would insert the needle, wait about 15 minutes for the tissue around the needle track to swell a little bit, so that to sort of close off the needle track, and then we would do an injection. Our assumption is that the inflammation in those conditions would be sort of minimal. We've also done a lot of injections with implanted guide cannulas, where you wait till the next day. But in those situations, you expect there to be a fair amount of neuroinflammation. We don't necessarily know what influence that inflammation is going to have. We suspect that it would have some influence. The problem is, yeah.
The other thing is, I've seen some papers in syringomyelia in this area, and I'm a little skeptical also because of the rate of infusion. Was your rate very low, or how do you know you're not increasing the interstitial pressure and that sort of causing an abnormal outflow or something?
Yeah, I think that's it. I think that's probably an issue. That's an issue that's definitely at play in this field, particularly, and we see it among other groups as well that have characterized these sorts of things. In these experiments, we're injecting 1 microliter over 5 minutes, so it's 200 nanoliters per minute. It's a fairly low rate. Even as you can see here, there's a lot of tracers. Where we gain a little bit of surety about things is the fact that when we look at longer time periods, so if we look 2 hours, 3 hours later, we see and we move posteriorly from here, we see that these patterns persist throughout the whole brain, pretty far distal from the injection site, and pretty late, so up to an hour or 3 hours later. So, and those are the time points where we would expect that any kind of focal increase in interstitial pressure that you would generate here will have dissipated by that time. But I think particularly, when you look at this cloud right here, I think most of this is probably driven by increased focal interstitial pressure increases. One thing that, and sort of going back to this question, one thing that would be nice to be able to do in an ideal world would be to find a way to deliver a tracer in an atraumatic way. So the other thing is we're producing also a low resistance outflow pathway here that is obviously not physiological. And so if there were some way to deliver a tracer into the brain without, putting a huge needle into the down through the cortical layers and probably disrupting this whole system in doing so, that would be preferable, but we haven't thought of a way to do that. Using maybe lower levels of infusion would also be good. What we run into is sort of when you get down to very low levels of infusion in the nanoliter range, you get into variability of just efficacy of injection. You get into issues of coring and other things like that, where the injections become much more unreliable at lower rates of infusion. So I think that's probably a pretty important point. So the next line of questions we wanted to pursue was the sort of brain-wide nature of this pathway. So all of this work was done in slice, so we lose dynamic information here. We were able to capture sort of the whole brain if we put enough slices together. but we see only one time. Maybe we can string 100 animals together, we can get a dynamic picture, but it's a pretty bad way to do it. The two-photon imaging gave us the ability to image dynamically, but we were restricted pretty much only to the dorsal cortical surface, so we were limited in terms of spatially of what we could do. So we wanted to try to get spatial and dynamic information at the same time. So with the help of collaborators at Brookhaven National Labs on Long Island, we conducted a dynamic contrast enhanced MRI study in rats, basically doing the same thing, but using MRI to image the whole brain CSF plexes, instead of using fluorescent tracers, using gadolinium-based contrast agents of different sizes. So we use Magnivist, 1 kilodalton gadolinium-based contrast, and we used a conjugated gadolinium called Gadospin that had a much larger molecule. We used the same route of injection and basically looked to see where the tracer went. And I don't know, is this movie come through at all?
Yeah, it looks great.
Okay, so this is the Gadospin. So this is a larger molecular weight tracer over time, and so we're injecting here into the cisterna magna, and what you see is that there's a very rapid brain-wide distribution of CSF anteriorly and then ultimately ventrally, and then you can see based on these dark guys here, it's perivascular, just like the fluorescence-based imaging showed. So you can see the tracer moving up along these, we presume, peri-arterial pathways, right? Along a similar distribution, but it seems to be brain-wide. Now, this tracer, you don't see a lot of parenchymal involvement. We assume that that's in part because of the molecular weight of this tracer, sort of trapping it in those perivascular spaces. When we use the smaller molecular weight Magnivist, you see much more parenchymal involvement. You lose some of the perivascular nature of it because it's not being trapped in that space. But again, with this method, there's a large portion of the parenchyma that is sort of signal change silent. We assume because we're just not able to accumulate enough of the contrast into the tissue to affect an MR signal change. So there's a sensitivity issue at work, we assume here. But the nice thing about this approach is we're able to capture sort of the dynamics and the brain-wide nature of this CSF interstitial fluid exchange pathway. So then
Jeff,
that leaves. Yeah, go ahead.
Jeff, sorry, I'm interjecting. So I know Vartan has done a simulation looking at tracers around the brain and just about that exact same, well, that region, right? Mm-hmm. I wonder, on your axis of time, mm-hmm. on that plot, was it 4.1 minute intervals?
What was each the scans were completed in about 4 minutes or so. And so basically every frame is 4.1 minutes, and then this is 61 frames. So this is 240. So this is a 4-hour imaging session. So this isn't real time, obviously. It's significantly compressed. But every frame is a 4-minute epoch.
So it's like at the beginning there, I don't see any dimension on this, but what's the size of that? Well, how big is it from the left to the right?
Oh, I see.
The distance.
So, yeah, I'd have to think about that. From here to here, you're talking about probably 2 or 3 centimeters. So from the back of the cerebellum to the front of the olfactory bulb.
So it looks like it's going about 1 centimeter in 4 minutes.
Yeah, so the movement along these perivascular, along these the large arteries of the circle of Willis and along the ventral surface of the brain is very rapid.
Okay. Really interesting. That's fascinating.
So then, where that sort of left us was with maybe a rethinking of the textbook version of what the CSF circulation is. So the textbook version is CSF secreted. It moves through bulk flow through the ventricular system into the subarachnoid space, from where it is absorbed one way or the other back into the peripheral blood circulation. But what this suggests is that a substantial portion of the CSF, at least the CSF within the subarachnoid space of the cisterns, is recirculating through the brain interstitium and potentially through the spinal cord interstitium. Our radio tracer experiments suggest that it's probably at least 40% of the CSF is transiting back through the brain parenchyma, and eventually we assume being cleared back into the subarachnoid space. So there's within this broader sort of loop of CSF secretion and absorption, there's this short circuit within it that's always operating, and we assume is subserving some sort of role in maintaining the brain interstitial environment and cleaning solutes out of it. Now, some questions that arise out of there is, so one thing is, we saw this arterial polarity of CSF inflow. So it seemed to move in along the arteries, but not necessarily along the veins. So what are the forces that might define this arterial polarity or drive CSF movements into the brain? The second question is, in between these inflow and these outflow pathways, there's a lot of cells. So there's neurons in between here, but more, I think importantly, there's astrocytes in here. And very insignificantly, these perivascular structures are entirely bounded by perivascular astrocytic influence and the glial limitans of the Virchow-Robin spaces. So astrocytes, these glial cells, sort of bound all of these spaces. And so do these cells have a role as in facilitating this movement or as gatekeepers of this movement? And then moving sort of from a clinical perspective, do changes in this CSF interstitial fluid exchange, does that contribute to neurodegenerative changes in the aging or the injured brain? So is there a clinical pathological significance of this system and its failure? So to begin to get at that first question about the mechanisms driving CSF inflow, in this field of CSF-ISF exchange, there's already an idea out there about arterial pulsation being one of the driving forces. This is a modeling paper from Ulbricht, who's at Cornell, and they provided a model explaining how arterial pulsation may drive perivascular fluid movement in the brain interstitium. Now, they're coming at it more from a convection-enhanced delivery perspective, looking at how drugs delivered into the interstitial space might move through perivascular pathways.
Oh, Jeff.
Yeah, go ahead.
Can I make a comment on that? So I haven't seen this paper, but there is a common misconception in this slide that you have, which is that the wave actually dilates the vessel locally. And this is not true. The wavelength is such that there wouldn't be a moving wave front that would move fluid forward because the wavelength is very long for arteries. So you see that slope there on the wave?
Yeah.
You wouldn't see that. It's the wavelength is like 5 meters.
In a human?
In human vasculature, the wavelength is very long. So basically all that there really is a wall moving back and forth. The vessel would be like a wall with no wave front per se to, for example, sort of milk fluid forward. This is very important. There was a paper published in Neurosurgery that talked about that, but it was actually since proven to be wrong. So I'm not sure what year this paper is, but maybe you have it interpreted in a different way, or I'm not sure if this paper said that, but this conception that there's a wave front moving the fluid it's just not true. It can't be. It's physically impossible.
This is something so this is sort of a biophysical question that we haven't had the expertise to pursue very far, and so we sort of have to take what's out there as from a modeling perspective. This person says this is a valid model. Oh, that sounds reasonable to us. That's a conversation I would love to have, particularly when we think about differences between mice and humans. So a mouse's heart rate, resting heart rate is between 300 and 500 beats per minute, which is a lot different than a human's. And so that's I would imagine, going to change the wavelength that we're talking about, right?
It would, but actually, Vartan, maybe you can give a little more detail. But it's, the wavelength is 5 meters, so it's basically a wall that has no slope to it.
Yeah, I'm not sure. I'm not sure if difference between mice and humans would make such a big difference in this case. There will be differences, but in this case, I would think that what Bern is saying is actually right.
Okay.
The important fact is that, at least in terms of pulse wave velocity, that's what's dictating this wave front, and the pulse wave velocity in the mouse or the human is basically identical.
Okay.
So this is not physically possible to have a wave front, milking, kind of like pushing fluid forward, but it can alter the hydraulic impedance in the perivascular spaces, the movement of these vessels, which is definitely, possible. So I'm not saying the whole concept is wrong. I'm just saying the concept that there's a wave front that could, traveling waves that sort of pull the fluid forward, that's probably not very likely with the wavelength being so big.
One of the questions we have is whether or not pulsation in this space, whether just from the vessel translating or from any source, and not necessarily peristaltic motion, is capable of producing, with the presence of a bound right here, pretend this is the astroglial end foot processes. Now, this is going to be somewhat flexible, but we can also regard it as perhaps somewhat rigid. Is that movement, whatever its nature, capable of producing hydrostatic pressure? And if so, is that capable of producing a pressure gradient from superficial layers down to deeper layers? And that's sort of something we would be interested in trying to get at from a modeling perspective. That biophysical level of understanding of these movements is not something that we've had an ability to get at. So our thinking, though, is that in one way or another, and we're not necessarily wed to this idea of peristalsis, pulsation itself is maybe one of the driving factors. So one of the things that we can do with the intravital two-photon microscopy, so we can image the vasculature, all right? We can isolate certain segments of the vessel. So this is a surface artery. We can also isolate penetrating vessels as well. We can conduct very high frequency line scans of the vessel wall, of the vessel itself. And what that ends up producing are these XT plots where you can you see the vessel diameter here dynamically with time being the axis here. So this is this single line projected through time. We can determine the diameter of this vessel as it over time. And what we invariably see in these vessels is that the diameter of the wall is in fact changing in a pulsatile way. So we see that the diameter here is deflecting, in the order of half a micron in either direction in time with the cardiac cycle. So we can observe this directly in the surface vessels. We can see this directly. This is a penetrating arterial here. We can measure basically the same thing. So when we think of pulsatility, we assume that whatever is the nature of this deflection, as the vessel gets larger in diameter and smaller in diameter, larger and smaller, the larger that deflection, and the larger the change between dilated versus constricted state with the cardiac cycle, that is going to have an influence on how fluid is moving along it. When we measure pulsatility along the vascular tree, along surface arteries, penetrating arteries, ascending veins, and surface veins, we see, as we measure it here, fairly substantial differences between the different pieces of the vasculature. So the surface arteries actually appear to have less of this sort of pulsatility than do the penetrating arteries and the ascending veins and then the surface veins. So the greatest pulsatility, as defined here, as sort of the integral the absolute value interval of these waveforms integrated around a moving average seems to be greatest along the penetrating arteries. We see the greatest deflections in vessel diameter there. We assume that that has to do with sort of thinness of the vascular wall of these penetrating arteries compared to the surface arteries. We assume that that is influenced by pulse wave pressure in these different levels of the vasculature. We assume that that may have to do with sort of surrounding tissue. We're not really sure what all the contributors are to that, but at least as we measure, we see greater pulsatility in these smaller penetrating vessels than in the surface arteries. Do you have any thoughts on that, Ben?
Well, sorry, I keep on interjecting. Does anybody else have comments? I have a question. Can you measure this at 2 locations at the same time?
We could. In principle, you could either do a single line scan between 2 vessels, right? So we could draw a line that went like this, right? Or at the surface, you could draw a line that kind of went like that, or maybe across these 2 where you can catch an artery and a vein. There's now some imaging capacity where we could draw more than an arbitrary line and conduct high-frequency scans at more than 1 point. So we now have more of a capacity to do that than we did before. These studies are all single.
You should measure the wave speed from that. You know, if you can do it at 2 locations and then do it as a function of, for example, the arterial diameter, you could plot wave speed as a function of diameter. That would be extremely interesting in these vessels. You know, what is the propagation of the wave in those tiny vessels? And also, one thought here is, in your technique, how much would the bulk tissue motion introduce error?
Yeah, that's a really good point. So we don't specifically have an answer to that question. What we do have is, we know that when you do these this is through craniotomy. The brain is it's pulsing within this craniotomy. You can do these studies where you put an agnos in this space that's opaque, or that you can image through, and that can reduce both the herniation and sort of the pulsation. But we can't necessarily rule out that this isn't just the brain pulsing with, the cardiac cycle, and then that is itself causing this artifactual movement. But our assumption is that it's not.
Jeff?
Yeah.
Zlatan, independent of whether it's the vessels that are moving or pulsating, or if it's the brain motion, would it be possible to do, let's say, external driving of the cardiac circuit in the rat to avoid pulsation?
Yeah.
And see if you can still get the same results. In which case you could exclude or say that it's the pulsation that causes the distribution of the tracers.
Yeah. So what we've tried, we've tried to modulate pulsation up and down through different interventions, so pharmacological and surgical. I think the ideal situation would be to, you put the animals on some sort of bypass, right, where you can create non-pulsatile versus a defined pulsatile flow, where you can really define its parameters. That's not something we've pursued. It's probably not something we have an intent to pursue right now, but I think it's something that we could do.
Yeah, that would be very interesting.
That's extremely interesting, Jeff. Have you ever seen it in that small of a vessel before? Anybody out there? I've been thinking of techniques to look at diameter changes. The vascular rarefaction in vessels smaller than 100 microns or 50 microns, I don't think has ever been quantified.
So the next goal was to try to change that pulsatility experimentally. So our first approach was the surgical one, and this is sort of in line with what other people have done with aortic banding or ligation of arteries, in hopes that by reducing either banding the aorta to prevent the propagation of the waveform through the vessel and render that flow somewhat non-pulsatile is kind of the idea, or by ligating the kind of the proximal circulation, you can reduce the exposure of your vasculature to pulsation, right? So that's kind of the idea. So what we did is we did a unilateral internal carotid artery ligation. So this is one of the 4 vessels that feeds the circle of Willis. We're helped a little bit here by the fact that the mice that we're using, the C57 black 6 mice, they actually don't have a complete circle of Willis, so they lack a posterior communicating artery. So the circulation of one side versus the circulation of the other, they're more isolated from each other than they are in other strains of animals. And so they're basically only linked anteriorly. So we're hoping that through this model we can sort of get some sort of vascular isolation and affect these flows. So here with this unilateral carotid artery ligation, and these are values taken 30 minutes after ligation, we see that there's a reduction in measured pulsatility in the penetrating arterioles. We don't really see any differences in any of the other at any other level of the vasculature. Not in the surface arteries, not in the veins, only in these penetrating arterioles. When we image CSF-ISF exchange with these animals, what we see is that these are in vivo results here. We see that the tracer injected into the cisterna magna moves along the surface and then down into the brain here. So this is 100, 160 microns below the surface. It accumulates at a rate, and we can quantify that here. After internal carotid artery ligation, you can just see visually, it seems to be slowed. And then when you quantify that, you can see that it seems to be statistically, it's significantly slowed by carotid artery ligation. Now, we don't have the ability to conclusively demonstrate it's the change at the surface vessels that is in fact changing this, right? So all we show is that the same intervention that changes pulsation in that compartment also changes this. So we just draw an inference. We assume that the two are in some way related. This is trying to go the other way. We wanted to increase pulsatility in those compartments. And so here we're using a pharmacological approach. I'm sorry, this is an ex vivo. We're basically doing the same thing with carotid artery ligation. So this is whole brain before carotid artery ligation or without it. This is whole brain after carotid artery ligation. And you see that there's more effect ipsilaterally. There's maybe some mild effect contralaterally. So this is basically the same data, but ex vivo instead of in vivo. So now we want to know if increasing arterial pulsatility makes this exchange faster. And so to do this, we used dobutamine, which is an adrenergic agonist, to increase cardiac contractility. Now, this had a similar effect, or had essentially the opposite effect of carotid artery ligation. It increased this pulsatility. A lot of this effect appears to be driven by a change in pulse rate. At 30 minutes after injection. But the dobutamine does seem to have an effect on this penetrating on pulsation penetrating vessels. And then you can see it has a correspondingly opposite effect on CSF-ISF exchange. This is ex vivo imaging control animals after cisternal injection. This is after dobutamine injection. So you see something similar going on. So that, to us, we think provides at least some evidence that that arterial pulsation is one of the forces driving peri-arterial CSF influx and exchange with the interstitial space. So the next question that we wanted to get at is whether or not, or what role cells that intervene between these pathways may play in facilitating this exchange. And we wanted to focus specifically on the astrocytes. So astrocytes, they extend end-foot processes that completely ensheath the brain microcirculation. So there's EM reconstruction studies that suggest that the ensheathment of a brain microvessel by end feet is actually complete, and that these are actually overlapping. So there's only a very small space in between adjacent end feet that would allow the movement of solutes out into the brain. So our natural assumption was that these end feet were sort of the gatekeeper for the movement of fluid and solutes out of these perivascular spaces. And that notion coincides with something that people in the glial field have recognized for a while, and that's this peculiar distribution of this channel called aquaporin 4. So aquaporins are they're water channels. The same way, they're similar to an ion channel, except that rather than conducting sodium or potassium, they specifically conduct water. Aquaporin is one of the two main aquaporin 4 is one of the two main aquaporins in the brain. Aquaporin 1 is present in the choroid plexus, where it's thought to be involved in CSF secretion. Aquaporin 4, when you look at its localization, it looks to be vascular based on this labeling. But when you look closer, this is immuno-EM done by a group in Oslo, what you see is that the aquaporin immunoreactivity is actually localized to the end-foot processes that face the microvasculature. So all of this purple that you see here is actually perivascular end-foot processes. And actually about 50% of the surface area of these hip feet that face the microvasculature are occupied by big plaques of aquaporin 4. So there's a huge amount of water conductance that's possible across this membrane facing the microvasculature. So our thinking is that maybe these water channels allow for the movement of fluid through the brain interstitium, and then from the interstitium back into the nervous spaces. So they would act as sort of a conductor for fluid movement. So to test that, we had available to us an aquaporin 4 global knockout mouse. So this mouse lacks a functional aquaporin 4 gene. So we conducted all the same imaging that I've shown you before on these mice. This is ex vivo imaging here. This is in vivo imaging here. And you see a profound failure of CSF interstitial fluid exchange in the knockout mice. So this is ex vivo. These are in vivo imaging. This is in vivo imaging conducted. These are frames taken from 120 microns below the cortical surface, 25 minutes after tracer injection. You see that in the wild-type mice, tracers in the perivascular space, big tracers, small tracers making it out into the surrounding interstitium. This is quantifying the movement of the small tracer into the interstitium here, so it's accumulating. In the aquaporin 4 knockout mice, you see that the big tracer is still able to get into the perivascular spaces. So those Burkow-Robin spaces are still patent, but the movement of the tracer out into the surrounding tissue is dramatically reduced in the knockout mice, and that's quantified here. So the absence of this water channel has a profound effect on this exchange. Looking at the other side, on the clearance of interstitial solutes out of the brain, we see that a similar effect of gene deletion. So in a wild-type animal, when you inject mannitol, which is a small molecular weight tracer, into the caudate nucleus, it's cleared from the brain at a certain rate. In the aquaporin 4 knockout animal, that clearance is reduced, which you just quantify here. The reduction is about 70% when measured at an hour. So there's a pretty big reduction in clearance of solutes out of the brain in these knockout animals. So that suggests to us that the aquaporin 4 dependent water transport through these astroglial networks facilitates fluid movement through this pathway and facilitates the exchange of CSF with the surrounding interstitial fluid. And that process, and in that process, also facilitates the clearance of solutes out of the brain interstitium. So that's sort of the mechanistic parts that I wanted to talk about. We're kind of running a little low on time, so I'll just mention briefly where our lab sort of goes from here is not necessarily delving deeper into these biomechanical aspects. These aren't things that we necessarily have a lot of expertise in. We're more focused on disease pathogenesis and in cellular neurobiology. So where we sort of go from here is toward whether or not failure of these cellular processes contributes to the onset of neurodegeneration or other pathological states. So just to highlight one, we look at microinfarctions. So microinfarctions are little tiny strips. They're usually less than a millimeter in diameter. They're present throughout the gray matter of the brain. They're fairly common among normal, non-demented elderly people, but you see a lot of them in patients with vascular dementia and Alzheimer's dementia. So they seem to be correlated with dementia. Whether they are specifically involved in cognitive kind is unclear, but they're definitely there. And so the thinking is that they're contributors to most forms of dementia. They're associated with atherosclerosis and cerebral amyloid angiopathy. Some people think that they're sort of the link between heart disease and neurodegeneration. So one of our thinking is that injuries like this, in addition to the focal injury that they cause, they could cause a more widespread model. We developed a mouse model of cortical microinfarcts, and in this model we inject cholesterol crystals that are filtered to be a certain size, so that they lodge deep in the penetrating cortical and striatal arteries. What you end up with are these little tiny infarcts scattered throughout the brain that themselves are very small. They're these little red guys here. These are all the little infarcts. But then, and that's quantified here in red. But then what's interesting is they're surrounded by these huge swaths of reactive astrogliosis, where the astrocytes have changed their phenotype to a reactive phenotype and are sort of in a defensive posture, right? So you have a small primary lesion surrounded by a wide area of neuroinflammation. What was interesting to us is that widespread neuroinflammation was associated with aquaporin 4 mislocalization. So under normal conditions, aquaporin 4 is expressed primarily along these perivascular entry processes. So it looks like it's a vascularly localized protein. But after injury, aquaporin expression actually goes up, but it ends up being mislocalized. You see it's not restricted to the perivascular entry. It actually is expressed along all the processes. And then that loss of polarization persists. Even this is 28 days after injury. This is a little microinfarct here. You see that loss of aquaporin 4 polarization has persisted out to 28 days post-injury. So there's a long-term mislocalization of aquaporin 4 after injury that has a consequence in terms of CSF, interstitial fluid exchange. So this is in vivo imaging taken from animals. This is 4 days after injury. You see that in a normal animal, CSF comes into the cortex, right? It peaks and then plateaus, and eventually it'll decline. In ipsilateral to those lesions, you see that there's a complete failure of that exchange process. And even contralateral to those injuries, you see that there's a reduction in that exchange. And you can see that in these ex vivo images. You can see under normal conditions, this is the extent of CSF-ISF exchange. In animals that have been subjected to those microinfarctions, and here you can actually see the lesions throughout the brain, you see that there's this global failure of CSF-ISF exchange. So that, to us, is sort of the main point that we want to focus on, and that we're focusing on moving forward. It's that when there is a diffuse reactive astrogliosis, what we almost always see is aquaporin 4 mislocalization. We see that after microinfarcts. We see it after mild and moderate traumatic brain injury. We see it in different models of aging. We see it in a number of experimental models that when astrocytes are reactive, aquaporin 4 becomes mislocalized, and this CSF interstitial fluid exchange becomes impaired. And so our framework moving forward conceptually is that impairment may contribute to the failure of interstitial volume clearance in those conditions, and then the misaggregation of different proteins like tau in neurofibrillary tangles, or like amyloid and amyloid plaques, or alpha-synuclein in Parkinson's disease, or other alpha-synucleinopathies. So our thinking is that this process of renal dysfunction and failure of fluid exchange may underlie a lot of different neurodegenerative diseases. So sort of what I'd like you guys to come away with from the talk is two main things, or three main things. One is that CSF circulation may not be strictly linear, a process of secretion, flow, followed by absorption. But what looks to be happening during that process, in that circuit, is that a substantial portion, maybe more than 40% of this subarachnoid CSF, is actually recirculating through the brain parenchyma. It's driven by arterial pulsation, and it's facilitated by astroglial water flux. So there's a recirculation of CSF through the brain parenchyma. Under different experimental conditions in aging brains, after microinfarction, after traumatic brain injury, we see that that recirculation is impaired. And so for your group and from your perspective, the bigger take home is that you perhaps need to regard the brain parenchyma as one pathway for CSF flow. So not just, when we think about CSF flow, don't just think about the ventricles and the aqueduct and the subarachnoid spaces and the arachnoid granulations and those sorts of things. But you perhaps should regard the perivascular spaces and then the surrounding interstitium as one of the pathways along which CSF is moving. So when there's a change in the parenchyma, specifically, and we kind of focus on changes in astrocyte phenotype, right, reactive astrocytes, that's going to affect broader CSF movement and the ability of CSF, through its exchange with the interstitial fluid, to participate in the clearance of different wastes from the brain. And so, and our view is that sort of failure may contribute to development of pathology in neurodegenerative state. So that's sort of what I wanted to tell you guys about. I need to acknowledge, I don't know if any of the folks are still here, the group in Rochester, that's where pretty much all of this work has been done. I'm just starting my research program in Oregon right now, but all of this was done in Maiken's group in her lab in Rochester. And she was my mentor and now my collaborator. Just a fantastic place to work. We had a pretty substantial team of folks who were and continue to be working on these projects in the group there. A couple of them, I think, joined us this morning, but all of them contributed to all of this work. Very little of it was, strictly speaking, actually my work directly. So thanks to them and to you guys. So that's it.
Thanks, Jeff, for taking the time to present this fascinating work. I think there's actually Frank Loth is here. He had a question for you regarding the direction of the flow in the spaces.
Hey, Jeff, that was an amazing talk. I really enjoyed it. Can you go back just a couple slides to your sort of takeaway points?
Sure.
I think it was maybe just one slide. Yeah, there. So facilitated by astroglial water flux. That was really interesting to me. I'm just wondering if that these astroglia, this flux, is it directional, or is it, like you were saying before, spherical in nature, going in all directions? It just enhances diffusion in general, or does it have a specific directionality to it that pumps it?
That's a super question. So the short answer is, and what we know is that we don't know. What we suppose, we don't know because water movement, particularly in the sort of microscopic realms that we're talking about, is very, very difficult to measure, and it's very difficult to follow and quantify. We're trying to think more about how you might do that, but at some point we just don't have the expertise to do the MRI studies or do the work that would be necessary to really know whether water movement per se is directional. Now, we think that a clue to that, though, resides in the expression of aquaporin 4. So aquaporin 4, you can see here, it's incredibly highly polarized, right? So the astrocytes are creating very specific domains for water movement. And those domains are between the perivascular spaces of the microcirculation and also the glia limitans at the cortical surface and in the Burkow-Robin spaces. And then the astrocytic network. So our assumption is that perivascular localization is involved in creating vectorial water flow somehow, that it's somehow moving fluid into and out of perivascular spaces in a specific way. One of the things that we see is that, and this is only correlative, this information, this tie, is that in conditions where that polarization fails, and you don't necessarily lose aquaporin expression. In fact, aquaporin expression seems to go up. But what it is it's expressed on all the fine processes all throughout the cell, right? So it's no longer polarized. What you see is a failure of these fluxes. So we assume that this polarization is critical to maintaining organization of these water fluxes and sort of keeping them going in a vectorial direction. And that loss of that causes you to lose that vectorial movement and causes you to lose sort of efficient movement. But the biomechanics of that and sort of demonstrating that ends up being very difficult, and we haven't really thought of a way to get at it yet. Does that answer your question?
Yeah, yeah, very much so. Maybe as a follow-up, on the arterial side, is it clearly directional in your mind? When I'm watching the videos, that really cool MRI video, it feels like the interstitial, the fluid is moving in the direction of arterial flow. Is that always the case, or is it most of the time the case? What's your intuition on that?
It seems to be almost always the case. And this is sort of the second level, right? So this isn't what you tell normal people. The flow, the rapid flow that we see is it's peri-arterial, and it's almost exclusively such. Right? We don't see rapid movement of tracer along veins. Now, when we say that, that gives the impression that we never see perivenous movement, and that's not true. We do see movement of tracer along veins, but it's slow. And to me, my take on it is that it looks to be sort of diffusion limited. So it's able to get into the perivascular, and you can actually see it. And you can see it early on here, right there. So there is tracer moving along these veins. You can see it here. There is tracer moving along these veins, but it's not being propelled, right? And so there seems to be a different sort of biomechanics at work. Now, whether that's because this is under arterial pulsation and these aren't, I don't know that that's necessarily the best reason. There's a group in Southampton, UK, who's done some morphological studies on cortical and leptomeningeal vessels and their ultrastructure that suggests that there may be an anatomical basis for this. So Roy Weller's group there, he's sort of moving toward retirement now. But they did a bunch of EM studies in human tissue back in the 90s, where they showed that surface arteries and veins are both surrounded by leptomeningeal sheaths, right? So they're both surrounded by peeled sheaths that follow. Actually, you can actually see it. That follow the vessels. In the case of arteries, those sheaths penetrate. I guess where could I show it? This would be the best. Here, no. Yeah, those sheaths are continuous with the Virchow-Robin space and move down, and then as the Virchow-Robin space terminates, they terminate. So the sheaths along the surfaces of the brain, along arteries, are continuous with the perivascular space of arteries. The sheaths along the veins, along surface veins, terminate at the peel surface, right? And so there's no direct communication between perivenous spaces on the surface and the Virchow-Robin spaces of penetrating veins. So there's an anatomical difference between those. And so, and those sheaths, I was reading those articles right when I was imaging this a couple of years back, and that exactly mimics what we see here, right? And that is, you see the tracer moving along what appears to be maybe a subpeel or some sort of subcompartment here, but then when it crosses over surface veins, it gains access to the veins, right? And our assumption is that it's doing so through the subpeel space there, right? And so it's not a strictly arterial versus venous movement, but that rapid conductive movement does seem to be very specific to these para-arterial spaces. That was a 10-minute answer to a 2-minute question, I think.
That gave me more than I was expecting. I do want to focus in on one thing though. So when the picture that you're showing me right there, this tracer is moving in the direction that blood flow is moving in the artery.
Always, yeah.
Always.
Always.
Now in the vein, so go back to that previous figure with the vein where you had a little bit of tracer for the vein. Yeah, this one right here. The venous flow is going the other direction, right? So is the
So the venous
flow
flow